Hello everyone, welcome to another video from DNA Bioscience. Today we are going to discuss about the BCR, that is called the B cell receptor. We have already discussed about the T cell, the type of T cell, that is the cytotoxic T cell and the helper T cell. And the glycoprotein which is present in the cytotoxic T cell is CD8 and that in T helper cell is CD4. And we have already discussed the T cell receptor. Now let's discuss about the B cell receptor. So before we start, let's discuss first about the lymphocytes. Lymphocytes are of two types. B lymphocyte, that is called B cell, and T lymphocyte, that is called T cell. We all know that B cells are produced in the bone marrow and the B cells are divided into memory cell and the plasma cell. The plasma cell is responsible for the production of the immunoglobulins, or you can say the antibodies. So let's discuss the characteristics of the BCR. It consists of two identical heavy chains and two identical light chains. These are two heavy chains and these are two light chains. The transmembrane region is present which anchor receptor in the plasma membrane. This is the transmembrane region which anchor the receptor in the plasma membrane. The next point is BCR binds to specific intact antigen. Then the B cell receptor for naïve mature B cells are membrane bound monomeric forms of IgD and IgM. So the antigen-binding site receptor is composed of membrane-bound immunoglobulin, that may be IgD or IgM. The disulfide links heterodimers called Ig alpha and Ig beta are present. You can see in this picture, this is the Ig alpha and this is the Ig beta heterodimer. The Ig alpha consists of a long cytoplasmic tail of 61 amino acids and the Ig beta consists of 48 amino acids. The alpha and beta are long enough to interact with the intracellular signaling molecules. So you can say the heterodimer, the alpha and beta chain, they are responsible for the signal transduction, or you can say this is the signal transduction region. So what are the differences between TCR and the BCR? TCR can only interact with antigenic epitopes that are represented within the antigen-binding cleft of MHC I or MHC II. But in case of BCR, they do not require antigen presentation with MHC. They can interact with epitopes on free antigens or with epitopes displaced on the surface of intact pathogens. The next point is TCRs only recognize protein epitopes, whereas BCR can recognize epitopes associated with different molecular masses. For example, the proteins, the polysaccharide and the lipopolysaccharide which is present in the gram-negative bacteria. So this is all about the BCR. Let's summarize this once again. BCR is the receptor found in the membrane of the B cell. B cells are produced in the bone marrow. They divided into memory cell and plasma cell. Plasma cell is responsible for the production of the antibodies, and the BCR structure consists of two heavy chains and two light chains. It consists of a heterodimer region with alpha and beta chain, and the long cytoplasmic tails helps in the signal transduction region. The B cell receptors for naïve mature B cells are membrane-bound monomeric forms of IgD or IgM. And the differences with the TCR is TCR require an antigen-presenting cell which first engulfs the antigen, chew off and crush up, then it represents the antigen with the MHC. But in case of BCR do not require antigen-presenting cell with the MHC. They can interact with the free epitopes. They can interact with epitopes on free antigen or with epitopes displayed on the surface of the intact pathogens. The next point is TCR only recognize protein epitopes. In case of TCR, the nature of the epitope is only protein. But in case of BCR, the epitopes are associated with different molecular masses such as proteins, polysaccharide and lipopolysaccharide. That's all for now. I hope it will help you. Thank you.